This is Dr. Mubin Sayed with one more episode of Long Story Short with Dr. Bean from the FLCCC platform. The discussion that we did last time was about nattokinase and its antithrombotic or fibrinolytic activity. Today we're going to talk about the anti-SARS CoV-2 or anti-spike RBD effect of nattokinase. These are in vitro studies. We do not have much in vivo data. And because of that, my own confidence about nattokinase for anti-SARS CoV-2 is not as much as the effect of nattokinase for being anti-clotting substance. So let's quickly look at the references first. So this is FLCCC or COVID-19criticalcare.com. If you go here, there are protocols, evidences, resources, news, webinars, education, and a lot more. This is how Natto looks like, this slimy structure here. This is the study that we'll discuss today. This is one of the studies, degradative effect of nattokinase on spike protein of SARS CoV-2. This is the other study that we'll look at. This is Natto extract, a Japanese fermented soybean food directly inhibits viral infections, including SARS CoV-2 in vitro. So keep that in mind. And also see, this is 2021. The other study that I showed you, that is 2022, August 2022. And that study, this study from August 2022 uses this study from 2021 or refers to that one. Then there are some more references in here as well. So let's start with our discussion. So quick summary, the question that we should have in our mind when we are discussing this is there sufficient evidence for anti-SARS CoV-2 activity within a human being or even within animals? So no, there is not sufficient evidence for that. There are in vitro studies that include human cells, but there are no in vivo studies. Then the second question will be, is it safe? So nattokinase or Natto, which has nattokinase as part of it, Natto has been used in Japan and the countries for thousands of years. So of course, in reasonable amounts, it is safe. So even for the in vitro studies that I showed you, and we would go over them, you will see that they took care to explain if the nattokinase that was helping to degrade the receptor binding domain of the spike protein or to resist the SARS-CoV-2 from entering a cell, that amount of nattokinase was cell safe or was not cytotoxic. However, there still is not sufficient evidence to say that, for example, we have other drugs that we have discussed in the past. There isn't enough evidence. There is one interesting thing though, and that is that nattokinase in these in vitro studies demonstrates that it can degrade the receptor binding domain of the spike protein. It is actually a serine protease. It is an enzyme, or it has an enzyme that breaks down serine part of the proteins, and serine protease are the active parts of the enzymes, and they help degrade or break down other proteins. Now let's start with our studies, and lots of good diagrams today. So here's the first study, degradative effect of nattokinase on spike protein of SARS CoV-2. So here the characters of this study, this is nattokinase. And why is it all slimy? Because it actually looks slimy. So it is all cute and nice thing, but it is slimy. So that is nattokinase or NK. Then you would see that in these studies, they use kind of a fluid that is similar in composition, not composition, but viscosity and properties to our serum. That is phosphate buffered solution. So we'll see that as well. Then there are two studies that we'll discuss. SARS-CoV-2 is used in one of the studies with the spike protein, and one study only used the spike protein. Actually, the study that used SARS-CoV-2, they also had bovine herpesvirus 1 as well. The reason they had two viruses is they wanted to prove that nattokinase can be antiviral in general. It can affect more than one kind of viruses. So the first study that I just showed you, what did they do in this study? What they did was they incubated nattokinase with cell cultures. What they had was, so let's say this is a set of cell cultures or a plate of cells with nattokinase on it. And then they had a 4-time dilution. So that means if it was, let's say, 8 microgram, then it was 4 times lesser than 2 microgram and so on. So these were the dilutions they used. The cells that were being used, these cells were S protein expressing cell lysates. What does that mean? These cells were transfected, they were changed to be able to express the spike protein on their surface. And then these were lysate, that means these were broken down cells. So that means in here where they put the nattokinase, this solution, if you will, contained broken cells, full cells, spike proteins, etc. Now they also did one more thing. They wanted to make sure that if the cell cultures in which nattokinase is present and let's say the spike protein breaks down, they wanted to make sure that that was nattokinase and not something else or not a fluke. So what they did was they also cultured more cells, but they put in them inactivated nattokinase. And what is the inactivated nattokinase? They would heat it, the nattokinase, at 100 degrees centigrade for 5 minutes. You would see that in the other study, they will heat it for 10 minutes. So I would then suggest that nattokinase heated at 100 degrees centigrade for 5 to 10 minutes will inactivate that. So this is how they will inactivate the nattokinase. And when they will put inactivated nattokinase on the cell cultures, then of course, the expectation is that if nattokinase is doing something, then inactivated nattokinase will not do it. So you could have this question that why will heat-treated nattokinase not work? So see, when we fry an egg, we treat it with heat and it becomes all coagulated and D-shaped, and the proteins inside do not work like normal proteins. Similarly, if you take other proteins and you heat them, we denature them, we deform them, they cannot go back to the normal state of functioning. So that is a point of heating proteins to test them. So imagine this heated part is actually inactivated nattokinase. Then they also did something else. They had the active nattokinase. They suspected that nattokinase had somehow in it a protease, that is protein degrading enzyme. They did not know, we actually still do not know exactly what part of nattokinase is a protein degrading enzyme, although from the research or from these experiments, we know that it has some parts in it that are protein degrading. So what they did was they put in another culture, they put protease inhibitor. They put another enzyme that breaks down or that stops a protease enzyme. And the protease enzyme is that they are suspecting is in the nattokinase. So they put something that could inhibit the active part of the nattokinase. So guess what? When the nattokinase, active nattokinase, was placed in the cultures, it was working according to the dose. So there is a relationship to the dose and the effect. So that is what was happening. When they placed in the same cultures inactivated nattokinase by heat, then it did not work. When they put a serine protease inhibitor, or just generally protease inhibitor, then also nattokinase did not work, which proved that nattokinase was active in degrading spike proteins, number one, and number two, if you inactivated it by serine proteases or protease inhibitors, the nattokinase becomes inactive. That means nattokinase has protease activity. So let's quickly look at the diagram. This is the diagram 1 in this study. So here, if I go to the diagram number 1, this diagram. So in this diagram, I'm just going to give you a quick review of how this is. Let's look at the B part. Okay, so in this B, look at this first column. So on the top, you are seeing that every column is nattokinase 1 microgram per milliliter for various time frames. So 0 minutes, 10 minutes, 30 minutes, 60, 120, 180, and so on. This PBS, phosphate buffered solution, is, you can call that as a control. Now the solutions, or these solutions, had nattokinase plus the lysate of the cells which were expressing spike proteins. And here if you see these bands, this is the Western blot. Or you can just say, let's say these are the X-rays for a general discussion, X-rays of the proteins. What happens is that the blots work in a way that there are negative and positive charges in the blot. Proteins have negative and positive charges as well. Plus protein, depending upon their size and charge, can travel from one anode, for example, to cathode. And how far do they go? They would reach a specific place and they would have a band there. So if you see here in the control first, S2 band is here and S1 band is here. Then if you see if the nattokinase treated cells are looked at, 0 minute, you see the bands. 10 minutes, you see the bands. 30 minutes, you see less S1 and S2. Why? Because nattokinase is breaking them. 60 minutes, there is almost no proteins. What happened? Nattokinase broke them. 120 minutes, gone. 180 minutes, gone. So that is what is important. And it was not a fluke because the PBS solution showed S1 and S2. 0-10 minutes also showed S1 and S2s. So that is the diagram here. If you see here cell lysate expressing S proteins, once again, similar thing. I think that we've made our point here. So going back to the discussion, the next thing within the same study, what is on the spike protein that is specifically getting broken down by nattokinase? So what they did was they had another cell culture. These cells were expressing spike proteins and they cultured these cells which were transfected to express spike proteins. They cultured them for 9 hours with nattokinase. So these were HEK 293 or human embryonic kidney cells 293. And what they found was that in a dose-dependent manner, if there was more nattokinase, for example 25 microgram per milliliter, then more of the spike protein from the surface of the cell was removed. That means nattokinase kind of munched it out and degraded it. If there was less nattokinase, then there was more spike protein left on the surface. That means it was the nattokinase that was working to degrade the spike protein. And so if you give it more time, it would degrade more. This is interesting if there is in vivo experimentation. This will be interesting for any possibility of a thought that, let's say there is messenger RNA vaccine which may be causing the spike protein being produced and expressed on the surface of the cells. In that case, this is the experiment that shows in vitro that nattokinase can remove the spikes on the cell surfaces. So that's very interesting. And let's look at this diagram as well. So if I go to diagram number 2, so here, this is the PBS, that is normal, let's call it control, phosphate buffer solution. This is 25 microgram per milliliter nattokinase. So here, compared to the nucleus volume, the amount of these red, the surface spike proteins was less. If the nattokinase amount was less, let's say dose was 2.5 microgram per milliliter, then the spike proteins were relatively more. So that was a dose-dependent effect on the cell surface expressed spike proteins. In this process, the cell stayed safe. That is very important. So then they say, in this study, we showed that the protease activity of nattokinase contributes to the degradation of S protein. Nattokinase has a degrading effect on not only spike protein, but also ACE2 in host cells. So this is why I have less confidence that it attacks the ACE2 itself as well. The protease specificity of nattokinase would be low. This is another concerning message, that the protease mean acting as an enzyme to break down a protein. And they're saying nattokinase acts as a protease. It breaks down proteins. However, it is not very specific for spike protein only. It was seen, just like with the ACE2, it was also seen or observed to break down other proteins. For example, GAPDH, a housekeeping protein, was also degraded simultaneously in the in vitro evaluation of nattokinase mixed with cell lysate. That is important to notice because in vivo, we do not want nattokinase and go and start breaking down other simple housekeeping type of proteins that are necessary for us. But please also remember, nattokinase has been used as a food item for thousands of years. So what is exact dose? What is exact behavior? We still need to have more studies. Then they say, on the other hand, when added to cells, it does not show any effect on cell viability and is expected to act as a protective agent on the cell surface. This is beautiful. Imagine like a window washer, right? Nattokinase is standing outside the cells and kind of washing them, removing the junk that is attached on the outside of the cell. The question is, are they going to remove the good proteins as well? But we are seeing here that at least the bad proteins are removed. So then they say further analysis of the degradation products of nattokinase using mass spectrometry is needed for understanding the proteolysis effect. So they say do more research. So this is one study, beautiful study. Next, this study is the September 2021 study. In here they said, natto extract, a Japanese fermented soybean food, directly inhibits viral infections including SARS CoV-2 in vitro. So again, the same characters. Here is what they did in this study. What they did was they had the PBS, the phosphate buffered solution, or let's say our saline type solution or serum composition, not composition, properties, viscosity. And then they have nattokinase. They put them together and centrifuge them. So they have the process, centrifuge them, and then the result is they take out the nattokinase extract from it. Then what they do is they take that extract, and as you can expect, they heat it for 100 degrees centigrades. So they keep some extract as active, and then they keep some more extract as heated at 100 degrees centigrade for 10 minutes. That means kind of inactivated extract. But they do not know if it is inactivated or not. They would just use it and see if it works or not. If it doesn't work, then they'll become sure that the active nattokinase is doing the function that they are experiencing or seeing or observing. So two types of extracts. Now what did they do? What they did was they have nattokinase extract cultured with cells plus viruses. So one set of plates had SARS CoV-2 on them with the cells and nattokinase. Another set of plates had the bovine herpesvirus 1 with nattokinase and the cells. They also had the same setup, but the nattokinase was inactivated. They also had one more setup without the viruses, and that was just the cells and the nattokinase. Why did they do this? They wanted to make sure that the cells that are being destroyed by the viruses were not being destroyed by the nattokinase. So this setup, where there was no virus, just the cell and the nattokinase, would demonstrate if there is any toxicity or cytotoxicity by the nattokinase. And I can just tell you right now that the nattokinase and the cell cultures did not have any cytotoxicity. So it was safe in the doses they used. So here what they did was 90 microliter extract, both types, and then 10 microliter virus, 37 degrees centigrade, 1 hour cultured together. Then they started diluting them. Then they cultured them for 3 days at 37 degrees. What did they find? They also did one more thing, and that is they took the receptor binding domain of the spike, just the receptor binding domain. And they also cultured it with both kinds of nattokinases, the heat inactivated and active. I did not make the heat inactivated, imagine that is here. And they cultured them together, 4.5 milliliter of each kind of nattokinase extract, and then 2 microliter, did I say milliliter here? Microliter. 2 microliter of the receptor binding domain of the SARS-CoV-2, then 37 degrees centigrade for 1 hour, they cultured them together. What did they find? I think you can now expect what would happen. So in a dose-dependent manner, the proteases that are present in the nattokinase were breaking down the receptor binding domain. Similarly, the nattokinase was inhibiting the SARS-CoV-2 from entering into the cell. So then the question became, how was nattokinase stopping the virus SARS-CoV-2 from entering the cell? And it turned out that nattokinase was able to degrade the receptor binding domain, so it would actually essentially remove the hand attached to the spike protein with which SARS-CoV-2 grabs ACE2. So that hand is trimmed or cut off, chopped off by nattokinase, degraded. And so the virus is present there, but it doesn't have the hand to bind to ACE2, and that was the interesting thing they discovered. So receptor binding domain, in one of the studies when they cultured the receptor binding domain with the nattokinase extract, that extract that was active in their receptor binding domain got destroyed. The inactive or heat inactivated nattokinase extract, their receptor binding domain was not destroyed. Similarly, on the plates where there was active nattokinase, SARS-CoV-2 and the bovine herpesvirus 1 were not able to enter the cells. And in the inactivated nattokinase, they were able to enter the cells. So this kind of proved how nattokinase works. So you can now see the summary in your, I hope in your mind, that number one, nattokinase attacks specifically or more the RBD of the spike protein. Secondly, it attacks it even when the spike protein is bound to ACE2 or is expressed on the cell surface without damaging the cell itself. However, there is a concern that how broad is its proteolytic activity that needs to be researched more. So then this is the conclusion of this study. They say, in this study we showed that the natto extract inhibited infectious ability of BHV, bovine herpesvirus 1, and SARS-CoV-2 to the cells and degraded the glycoprotein D of BHV1, that is the receptor over there, and RBD part of the SARS-CoV-2, that is the receptor binding domain here. However, this inhibitory effect of the natto extract was impaired upon heating as well as treating with the serine protease inhibitor, which proves that nattokinase has these active parts in it. Hence, it is conceivable that this inhibitory effect was due to the putative heat-sensitive serine proteases. This is further supported by the evidence that the protease was able to digest multi-sites of glycoprotein D and RBD protein. It could damage those binding units of the viruses. Therefore, our study suggests that the putative serine protease in natto may impair the infectious function of BHV1 and SARS-CoV-2, probably through the proteolysis of its glycoprotein D, that is the receptor binding area of the BHV, and the spike protein, that is SARS-CoV-2. Further research must focus on identifying the proteases and locating the cleavage sites. Isn't that interesting? So they say that we are observing nattokinase doing this, but what area within the nattokinase is exactly the protease? Where is the cleavage site where the breakdown, that protease causes the breakdown of the spike protein or the other virus spikes? That needs to be figured out. Purified proteases in the natto extract is worth to be further investigated as antiviral drugs. Finally, our findings open a future avenue towards new applications of natto as raw material. So, beautiful product. I think it is still not sufficient to say that this can prevent COVID or this can help, but it is safe enough and promising enough that further studies can be done, or if it is being taken in the food, one can probably attach some hope with it as well. But on the other hand, the blood clotting or fibrinolytic activity has much more evidence for this. So with this, thank you very much. Happy holidays, and I would see you next time.